I'm delighted that so many of you are able to stay for the day and to hear this talk, which I know is going to be, I'm really looking forward to, and I heard a preview of it because earlier. So it's Eric Hollander is professor of psychiatry and behavioral sciences here at Einstein and director of the Autism and Obsessive Compulsive Spectrum program and anxiety and depression program at Montefiore. He has a clinical practice as well, and he's involved in a lot of clinical trials research on the neuropharmacology and treatment of symptoms in different neuropsychiatric and neurodevelopmental conditions, including obsessive compulsive disorder, Prader-Willi syndrome, autism, and body dysmorphic disorder. Eric is also a key member of our new Psychiatric Research Institute of Montefiore Einstein, also known as PRIME. And so,
okay, well, thank you, Sophie, and the organizers, both for that kind introduction and for inviting me to participate today. It's really been a great conference. So I'm going to talk a little about experimental therapeutics, mostly looking at oxytocin and vasopressin and also the cannabinoid system, and focusing on core dimensions like social communication and repetitive behaviors and associated domains like irritability as well. Some of our work has been funded by the Department of Defense for the CBDV study in autism and Orphan Products Division of the Food and Drug Administration for oxytocin and Prader-Willi. And I'd also disclose that we're working with GW Pharma for our work with CBDV and autism and Prader-Willi, and we've been working with Roche to develop a vasopressin 1A antagonist for autism and to develop biomarkers for the repetitive behavior domain. So here's our team at the Autism and OC Spectrum program. Bonnie Taylor's been playing a key role in the oxytocin studies, and Vera Nezgo-Varova for the CBDV studies. We've also been working with Oren Davinsky at NYU. So here's our book on autism spectrum disorders. So it's a nice overview of autism, both for clinicians and investigators in the field. And so here's sort of the core and associated symptom domains. As you know, in DSM-5, the social and communication domains were combined together into a social communication domain, and we have repetitive behaviors. The impulsivity and aggression is this sort of irritability or disruptive behavior domain. And we've been hearing about high comorbidity with EEG and epileptiform activity and a lot of ADHD symptoms as associated symptoms. When kids come in for treatment, they come in for different kinds of symptoms at different stages in their development. So early on, they may not be talking, or they may have self-injury. Later on, they may not be interacting with their peers or have physical symptoms. The rigidity may cause a lot of discomfort, and they may be exploding. Later on, they may develop social anxiety or depression, and later on, they can have relationship problems or workplace issues or develop impulse control problems like internet addiction. So the current or standard treatments that are out there from a medication standpoint to treat some of these different domains are things like risperidone and aripiprazole, which have an FDA approval to treat the irritability or disruptive behavior domain. They really have a large effect size, but they are associated with a lot of side effects. So weight gain and metabolic issues. We're participating in a multi-center study to try to measure the impact over time, particularly on metabolism as a long-term adverse event for the atypicals. So other treatments are needed. We did some studies looking at fluoxetine to target repetitive behaviors, and we found both in children and in adults that we actually got a reduction in the severity of repetitive behaviors with SSRI. Helen was also involved in the START centers, And as part of that initiative, we did a multi-center citalopram trial, which really showed no difference between the active medicine and placebo. So there was a huge placebo response rate, and we wanted to try to understand that. So we subsequently went back to try to stratify the population and just select those individuals who had high irritability on the ABC irritability. And if we looked at those individuals who were having frequent episodes of explosion, the placebo response rate dropped from about 50% down to 5%. And in that population with frequent episodes, we actually had a nice separation of drug and placebo. We did some of the early studies with valproate, an anticonvulsant, and it did seem to have a nice effect on irritability. And we also noted that those individuals that were having epileptiform kind of discharges in particular had a nice response. But there are some sort of acute and long-term side effects also associated with valproate. And then finally, stimulants are often used to target inattention and motor hyperactivity. And they also can have some side effects. And those side effects are probably higher in the ASD population than in the ADHD population. So one idea is to try to get more homogeneous subgroups to try to decrease this placebo response rate or get more homogeneity. So you can stratify based on higher target symptoms of the domain that you're going after. And Naburo talked about a dimensional approach to different domains. Or you can try to get more homogeneous orphan syndromes where there's a common underlying genetic mechanism, and then try to match the compound to the underlying molecular mechanism. Then finally, there are issues in terms of our outcome measures. And so another issue is to develop biomarkers that are closer to the underlying mechanism of the illness. So for example, we're working with Roche to develop better measures for repetitive behaviors and things like rigidity, which hadn't previously been measured, and then correlating all kinds of different measures of rigidity with a broad range of different kinds of biomarkers as well. So I'm going to talk a little about some of the work with oxytocin and vasopressin and a little bit of the work for CBDV, targeting things like irritability, repetitive behaviors, and social functioning. So oxytocin is a peptide that's made in the hypothalamus, and then it's excreted into the bloodstream through the pituitary, posterior pituitary. Peripherally, it has all kinds of effects on metabolism, milk letdown, labor and delivery, lactation. Centrally, it has a lot of effects on things like social recognition, social memory, trust. So this also is a G protein coupled receptor that's linked to the PI3K ACT mTOR pathway. There's a lot of communication between the peripheral and central oxytocin system. And the vasopressin system and the oxytocin system seem to work with reciprocal kind of functions. So you can think of oxytocin as stepping on social gas pedal and vasopressin as being sort of a social brake, for example. But these things are also involved in lots of other effects. So things like wound healing, they have very potent anti-inflammatory effects, and they're very much involved in things like metabolism and obesity. So if you knock out the gene for oxytocin, then mice may have a hard time recognizing other mice. Or if you look across species, those species that have a lot more oxytocin and vasopressin receptors in the social brain seem to be much more affiliative and have long-term sort of partner bonding. And those species that have much less vasopressin or oxytocin receptors in the social brain may be more aloof or less affiliative. You can administer oxytocin by a saline and oxytocin nasal spray, and you can enhance things like trust in healthy volunteers through economic type models. And then you can measure amygdala reactivity as sort of a measure of social threat. So you can show people angry faces or fearful faces. And normally individuals light up the amygdala in response to social threat. and giving them intranasal oxytocin dampens down that response. We can administer oxytocin through an IV, for example, and get a reduction in repetitive behaviors, particularly these sort of lower order self-stim type repetitive behaviors. So what we call the lower order as opposed to the more higher order compulsive anxiety-driven repetitive behaviors. We developed a measure to try to tap into social cognition, the ability to recognize emotions. So we just read a neutral sentence. The boy went to the store, initially with 4 tones: happy, indifferent, angry, or sad. And then we infuse oxytocin intravenous, or we infuse placebo over a 4-hour time period. And then we repeated this affective speech recognition every hour. And we found that both on the oxytocin and the placebo, with a little bit of practice, individuals who were high-functioning young adults with autism were better at recognizing these emotions. What was interesting, though, is when they came back 2 weeks later, those people who had gotten the placebo initially lost their expertise in recognizing those emotions. So those people who had gotten the oxytocin initially, maintained that expertise. So there was this drug by time by order effect, and we thought about this for a while and thought, well, what was going on was that there was some social learning going on with the repeated task on the oxytocin arm, and that persisted over time. They were laying down social memories. Then we made the task a little bit more complicated. We added some tougher emotions like lust and fear. So the boy went to the store, but in a lusty or a fearful voice. And then we administered one of these vasopressin 1A compounds that Roche was developing, an antagonist that specifically blocks those receptors. And we were surprised to see a very large effect size, 0.7 or 0.8, on the ability for these high-functioning young adults to recognize fear and lust. Well, it was going in the negative direction, which was surprising. So they were actually getting worse at their ability to recognize fear or lust. But then what was interesting was that the autistic individuals were actually better than the typically developing individuals and recognize them. They're very, very sensitive to fear and lust, and then they come back towards the norm in response to the vasopressin 1A antagonist. So then Roche developed a longer-acting vasopressin 1A antagonist, balovaptan, and they did studies first in adults and then adolescents, and we're continuing to do the studies in adolescents and now doing large-scale studies in adults. They looked at the outcome on the Vineland, and after a 12-week time course, what they saw was a significant improvement, particularly on the higher dose, this 10 milligram of the vasopressin 1A antagonist, specific only on the social communication domain, and at the 10 milligram dose, a kind of a moderate effect size of almost 0.5. So this is very different than treating irritability where people are exploding and you're trying to decrease a behavior. This is something where people don't have it at baseline and you're trying to build it from baseline. It's a very different kind of a thing. Because of this, the FDA gave a FDA breakthrough designation, and they're going towards sort of pivotal trials now. So then we thought, well, one way to try to reduce heterogeneity is pick rarer syndromes like Prader-Willi syndrome. So all of these individuals don't express paternal genes in this region, 15q11 to q13, but there are different subtypes. And these kids start off, they don't eat, and then all of a sudden they have this craving for food and they become highly obese. They are obsessed about the timing of meals, and if there's any deviation, they experience a lot of discomfort, and then they explode. They have temper tantrums. They have a lot of compulsive and skin-picking behaviors, mild intellectual disability, but some social cognition deficits. They also have a decrease in the number and size of oxytocin-producing neurons in the brain on autopsy. And mice that have decreased oxytocin neuron activity have obesity, and then they can be treated with oxytocin to decrease their body weight and food intake. So one question is, does the intranasal or the intravenous oxytocin get into the brain? Well, we can measure it in the spinal fluid, and after they get it nasally, there's an increase in the spinal fluid in rhesus monkeys or peripherally, also the same intravenously. That doesn't tell you the long-term binding, and there aren't good ligands. But it's led to a study that we have ongoing now in Prader-Willi syndrome. So 50 kids, intranasal oxytocin or placebo, where we're looking at hyperphagia over a 3-year study. We also are looking at repetitive behaviors, weight and BMI, body composition, things like the relationship between salivary oxytocin and then safety-related issues. We want to kind of integrate some biomarkers as well. I had talked about placebo response as being a challenge in autism and even in rare conditions. And that placebo response can be parceled to an expectation bias, which can be blocked by opiate antagonists and a social learning component. And biomarkers may be able to address those kind of issues. Well, we did some studies with Tricura suis ova, which is it's the egg of a pig whipworm, which had been used to treat some inflammatory bowel disorders and other autoimmune type illnesses. And we wanted to see whether it had any impact in autism, and we were particularly interested in the repetitive behavior domain. We developed a scale called this Montefiore Einstein rigidity scale, which gets at how much in response to deviation from expectation people experience discomfort and then protest. That's not really picked up with the current scales. And one of the things that we saw was a large effect size in terms of rigidity on this sort of anti-inflammatory kind of treatment versus placebo. And then recently there's been a lot of interest about the blunt truth of weed and autism. So it turns out that across the country, families are actually giving their kids cannabis, particularly to treat some of the explosive behaviors that we see in these individuals. Because when they explode, it causes a lot of caregiver stress, and it limits the ability to go out into the world as well. So we've been interested in looking at a CBDV, cannabidivarin, and we got some funding by the Department of Defense. It impacts military readiness. If kids are exploding, then they can't be on certain military bases where they need behavioral kind of treatment. So it does have impact on things like military readiness. So CBDV has opposite or reciprocal effects to THC. It really doesn't have any THC, which is the component in cannabis that gets you high. It has less than 0.2%. It doesn't bind to these endogenous cannabinoid receptors, CB1 or CB2. It binds to this G protein receptor 55 and to this TRP receptor, although this GPR 55 may eventually be called a cannabinoid 3 receptor. It has a potent anticonvulsant activity. So CBD just recently got approval for 2 rare forms of epilepsy, and it's about to come into the pharmacy and be marketed for epilepsy. This has interesting anticonvulsant effects and definitely decreases excitation relative to inhibition. It has pretty potent anti-inflammatory effects. It reverses aberrant synaptic plasticity and does have some antipsychotic or anti-addictive effects. So it's being looked at as a treatment in things like schizophrenia, for example, or addiction. So there's some evidence in autism and in Fragile X and other inflammatory disorders that the endocannabinoid system is involved. There are some animal models where they've given CBDV, so a tuberous sclerosis animal model where it decreases seizures or EI imbalance, Fragile X model where it enhances socialization, the valproate model where it has an impact on repetitive grooming, and a Rett model where it has some effects on enhancing cognition. And it's been studied in some patients with refractory epilepsy. So you have CB1 receptors in the central nervous system, CB2 receptors throughout the body that are primarily immune function, and the endocannabinoids, anandamide or the bliss chemical, and 2-AG, and then these phytocannabinoids, THC, CBD, and CBDV. So THC binds to these cannabinoid 1 and 2 receptors and activates PI3K, ACT, mTOR pathways. CBDV binds to the GPR55 and TRP channels, not to the CB1, CB2. And these cannabinoid mechanisms are found throughout the central nervous system in myeloid cells and in glial cells as well. So our study, we're looking at kids with autism. 100 children are going to be recruited here and at NYU, and kids 5 to 18, where we're going to titrate up to 400 milligrams twice a day in a randomized parallel design study. And then after they do the acute trial, these kids can come in for 6 months of open label treatment. So we dose up to this 10 milligrams per kilogram per day. They come in every 2 weeks. We're looking at irritability, so exploding with things like the aberrant behavior checklist, repetitive behaviors with the RBSR and the MERS, global functioning, and then it's important to look at things like liver functions and the interaction between this and other drugs, some of the anticonvulsants. So this will be the first study of a cannabinoid in autism. It avoids the effects of THC. It has a lot less potential adverse events than some of the metabolic adverse events associated with the atypicals. And we're doing some studies looking at adverse events of atypicals. It's not medical marijuana. Medical marijuana has been approved for autism in states like Rhode Island, and Autism Speaks is having a conference now to try to design effective trials to assess risks and benefits. The other thing is to incorporate what families and patients think are important kind of outcomes into these studies. So we've been focused on target domains. We've used orphan disorders to kind of get more homogeneity. We try to match compounds to underlying mechanisms. We try to understand more sources of placebo response and try to incorporate some biomarkers to try to drive that down, and then speak with families in terms of what they think are important outcomes as well. So thanks very much.
Let's take time for 1 or 2 questions for Eric, and then we'll have our final talk from Shlomi. Questions?
Eric, it's not so much about your interventions, but a question to somebody who's really got their finger on the pulse of interventions in ASD. Is there any news from the front in terms of neuromodulation types of work in ASD, and do you have any comments about that?
Yeah, so that is a active area now. We've been involved in some studies, and others have as well. So there are a couple of approaches to that. One is to take TMS protocols that have been used in related conditions, so like OCD. There are 2 approaches in OCD. So low frequency stimulation in Helen's area, this supplementary motor area, to decrease the activity and decrease the frontostriatal thalamic circuitry. And there have been some studies where there's some improvement in the repetitive behavior domain. Standard TMS, like dorsolateral prefrontal cortex, with low frequency also to decrease things like irritability and this excitation inhibition imbalance. And then deep TMS was just approved for obsessive compulsive disorder. That's high frequency stimulation in the anterior cingulate. So to borrow some of the protocols that are done in related conditions and apply it. In particular, 2 of the important strategies are to decrease this excitation inhibition imbalance and maybe to target repetitive behaviors.
I wondered, in your study on CBDV, are you looking at sleep as one of the confounding factors or one of the elements of irritability, because I know that CBD, general CBD, has an effect on sleep, and it might be something that you should consider.
Yeah, no, I think it's a good observation. You know, so things like CBD are out there. People can get it on the internet. They're using it for all kinds of things. And one of the reasons they're using this to enhance sleep and sleep architecture also. So it's not one of our primary or secondary outcome, but probably you're absolutely right. So sleep is a major unmet need, I guess, in the autism community. Yeah.
All right, thanks again, Eric.
Okay, great. Thanks.